And with this first, we'll be presenting Cole Smith. And Cole Smith will be presenting on the evaluation of certified organic fertilizers for long-term nutrient planning. Cole Smith is a staff research associate with the University of California Agriculture and Natural Resources in Santa Clara and Monterey counties. Cole's work focuses broadly on implementing extension-based research and education programs to increase adoption of sustainable production techniques. Cole specializes in composting education, organic waste amendment research, and community-based learning. Cole is also a graduate student in the soil science and biogeochemistry program at UC Davis, working under Dr. Will Horace. Welcome, Cole. Take it away.
Hello, everyone. Welcome, and I just want to say thank you to FREP and WPHA for inviting me and hosting such a great conference. And I just want to second that this meeting has been seamless and has flowed very well. Okay, so let's get started. Yeah, my name is Cole. I'm a research associate with both UCANR and UCD, so dual affiliations there. And we've been working on this project to evaluate organic fertilizers for long-term nutrient planning now for about 3 years. So I just want to share our project with you today and get some feedback. Okay, in this session, I want to go over critical elements of long-term nutrient planning. And with understanding long-term nutrient management planning, we need to understand soil organic matter and soil organic matter formation and how the nitrogen cycle overlaps with this process. Then I want to go through the factors that influence the nutrient release or nitrogen release from organic amendments and try to look at what factors are important for the short term and for the long term. I'll go over the current project results, and then we'll talk about where we're going with the project in the future. Okay, so when we think about long-term nutrient planning, we need to understand soil organic matter formation and cycling. So the nutrient reserves that we're going to build or destroy via soil organic matter planning are going to be critical to the long-term outcomes of our management. So in the past decade, we've seen a shift in soil organic matter studies. It's a very hot topic. Many people, many scientists are looking at it due to soil carbon sequestration. This shift has moved from understanding soil organic matter in the persistence of soil organic matter and nutrients bound up in soil organic matter from understanding it as a more recalcitrant, that long-term soil organic matter is more recalcitrant plant material, shifting to understanding it more as microbial derived material. So here you can see in the figure that our inputs here as either plant inputs, live plant inputs as root exudates or inputs from organic matter or fertilizer, anything really, is going to be intercepted by the microbial life cycle or microbial growth cycle. And as this microbial growth cycle turns over and recycles, a portion of the carbon that's consumed or nutrients that are consumed are respired, and a portion are sort of locked in or entombed into the microbial biomass or the microbial bodies and cellular matter. So you can see that this cycle turns over and you end up with dead microbial fragments. So this is cellular material and things, just the leftover residues that microbes produce. Those then become associated with minerals as well as organic matter existing in the soil already. And this process and the efficiency at which this process happens determines soil aggregation. So if you've heard from soil, any sort of soil health information, aggregation is a very important component of soil health, and it's a very important component of long-term organic nutrient persistence in the soil. So if we start to shift our thinking around not necessarily stocking the soil with recalcitrant plant material, but thinking about it in terms of feeding and driving this microbial cycle, then we can start to gain a better understanding at how long the amendments or the management will persist. So then these nutrients are stored in these soil aggregates, and the ability to either store those or lose those determines that long-term nutrient outcome. And microbial carbon is sort of the backbone to this nutrient storage. So when we are talking about organic fertilizers in particular, we should understand it as two types of controls on the release of nitrogen from these materials. So you have the internal controls, which are often sort of generalized as C to N ratio or carbon to nitrogen ratio. But really, this could also be a little more detailed than that. This could involve different carbon complexities or different total nitrogen amounts. But also, I wanted to look at external controls, because the internal controls might control the short-term release of the nutrients, but if we want to understand the long-term release of the nutrients, then the external controls could be a very important factor, so we'll review those. But this diagram here showing the release of nitrogen from these materials sort of shows how complicated this can be, and the reason that we're still unable to predict these outcomes in different soil environments. So we have this linear process of mineralization, releasing organic nitrogen into inorganic plant available nitrogen here. But this process of mineralization, although it seems and appears linear here, it's not so linear. So you have the process of a large organic pool, and a smaller portion of that organic pool is that microbial biomass pool. And the release of nitrogen from this pool is a process of it going from a complex form to simpler forms, finally then into the inorganic form. And this process can be very dynamic, meaning that it can cycle back on itself. So these organic, smaller molecule organic nitrogen forms can become available and then be consumed depending on management, or they can be released. The rate of release is dependent on many different factors. So this process is quite winding and not necessarily very straightforward. And that's why we're still here talking about this. So if we look at amendment characteristics, it's a very common way of viewing and assessing organic amendments or fertilizers. Using the total nitrogen is a very common measure of gauging and trying to predict the release, total release of nitrogen from these materials. And so also carbon complexity. So the common generalization is the C to N ratio, but this can also be a lignin to nitrogen ratio. Some researchers have looked at that. So it really depends, but this can be characterized as the C to N ratio. And here on the side, the common conception is that the C to N ratio narrows and we have a faster rate of decomposition. And so here it shows, I like this figure because of this, it is highlighting the microbial diet. So the C to N ratio of about 24 to 1, something around there is the microbial diet. And that's very important if we think about driving that microbial process, that trying to drive as much microbial material into the soil organic matter as we can. And so this microbial diet is also important because they're respiring half of their carbon that they're consuming. So if they don't have the carbon available, then they're not able to turn over that material. And so they need that carbon just like we need carbon for energy to grow. But they're going to lose half of that carbon, as that first figure showed, and respire half of that carbon. So this is sort of looking at C to N ratio in the very traditional way, and it looks like this has gotten cut off. This says immobilization and mineralization. And so we see as the C to N ratio narrows, often the material itself becomes more simple. So the heterogeneity of the material, the chemical complexity of the material can become simpler as well. That's not always the case, but it's often the case. But really, when we think about adding lower C to N ratio, we should consider that we're increasing that added organic nitrogen pool. And that added organic nitrogen pool changes in size, but it also changes in complexity. So oftentimes we're adding it further down that line of mineralization and further simplifying the form of nitrogen that we're putting down. And this, as this shows here, this can increase the amount that becomes plant available. So a larger percent of that pool of organic nitrogen then becomes available. So predicting this for growers is a huge challenge, and that's why we're here still researching it. Okay, so to look at some of those external controls, the major one under irrigation or precipitation and temperature changes throughout the season or in the future under warmer scenarios. So if we look at temperature, has a control on N release, but when we look at across different organic amendments, the temperature response is not always the same across organic amendments. That's what has been found in the past. And really, temperature can have a huge influence on the rate of release. And that can be really important when we're trying to time the plant uptake of nitrogen and the release of nitrogen, specifically over multiple seasons, or if it was an early season application and you have a second crop going in, for instance. So also moisture, but moisture hasn't been consistent either across with having a consistent effect across organic amendments and influencing nitrogen release. So these are important factors that still it's unclear what type of influence they have. So if we look at soil environments, we're looking at different things such as soil types, so edaphic characteristics of soil. So this can be as simple as different percent sand. So are you on a sandy soil or are you on a clay soil? And that percent sand can often shows to increase because there's more oxygen in the soil matrix. So the material can mineralize out quicker, decompose quicker. But with lower C to N ratio amendments, the work is limited, and we haven't necessarily concluded the effect from total sand. So that's the component of the mineral component of the soil. Now, if we look at the biological component of the soil, that can have a major influence, and it's important from this point. Just how I was talking about earlier with the microbial diets, is those microbial diets are fixed. The nutrient requirements generally are held pretty constant. So if you don't have one component of their nutrition, but you're loading the system with another component of the nutrition, it doesn't do the microbial community very much good if they're not having a balanced diet and they're not able to gain energy from the inputs. So that's important when we think about long-term soil organic matter building and building up these long-term organic nutrient reserves. Because it doesn't matter of the amendment that you apply, those microbial nutrient ratios are going to be fixed. So in the short term, we have organic amendments have shown to increase biological activity, can increase microbial size, so the whole size of that microbial community can increase. Also, the activity of the microbial community, such as the release of enzymes to break down that material, can be increased through organic matter additions. But when we look at the long term, we're really looking at changes in carbon and nitrogen pools. And a lot of organic amendments have shown, higher C to N ratio organic amendments have shown to increase carbon and nitrogen pools in the long run. But with the organic fertilizers that are lower in C to N ratio, this is still unclear, the long-term outcomes. Okay, first survey break. Yeah, it's interesting. We have quite an even distribution, it seems like, everybody's mixed with their responses. So that would be great to take a look at this deeper. Yeah, I see that moisture popped out there as an important characteristic of nutrient management planning. So hopefully we can get some questions answered or hear your feedback during the Q&A as well, because we're very interested also just to see on the, to hear what's happening on the ground in your decision making. Okay, let's keep going. So in this project, we took an approach of collecting data from the literature, conducting some laboratory incubations, trying to get some field measurements on how organic amendments influence the end cycle. And then eventually we want to get this into informing some models that will project our long-term 5 to 10 year, 10 to 20 year horizon outcomes from these management choices. So like I was saying with the organic nitrogen pool, really what we're most interested in is how susceptible that organic nitrogen pool is to mineralization, and being able to better understand from these materials how much of that organic nitrogen pool will then become available, either in that first season, second season, or in the long run. So this has been done by fitting different models to incubation data. And really it's driven by the question of how much how much nitrogen will become available in the inorganic form, and how fast is that going to become available. And that's described here by the N naught value as that potentially available pool, and then that specific rate constant is the turnover rate, or how quickly that's going to become available. And then what factors are determining that release. So in our project, here's some of the data, and so we've extracted data from 16 studies incubating compost and 5 studies incubating organic fertilizers. There's not as many studies on organic fertilizers as compost, but we wanted to make that comparison. So we've extracted compost, manure, and organic fertilizers. Here you can see on the right, just to orient you, this is just the model fits. So it's just the parameter values for organic fertilizers. So you have the K, which is the rate constant of release, and then you have that percent nitrogen available from the pool, and this is represented here on the x-axis as that percentage. So this is percentage on the x-axis and C to N ratio, and you can see here that as the C to N ratio tightens, you get that expected trend, that the amount that's going to be potentially available increases. So there is that trend, but you see here that the bars represent the uncertainty around these parameter values, and there is some overlap. So there is some uncertainty, but we found here with the organic fertilizers that you do get a pretty clear trend. But interestingly, you don't get that same trend with the rate constant. So the driving factor of how fast this nitrogen is becoming available is not as clear as it is with the potentially mineralizable N. So when we compare amendment types, so we're comparing compost and fertilizers here, organic fertilizers. So in general in our study, I coded it based on material, but they really did come out based on C to N ratio. So our organic fertilizers were under 10, whereas compost generally were over 10 C to N ratio. So we compare the potentially mineralizable N from compost and fertilizers, and as expected, you do have a significant difference between those two. So the mean value here is over 50% for the organic fertilizers, meaning that 50% of that organic nitrogen pool is going to become available. And then with compost, you have less than 20% of that, showing that it's going to become, the mean value is less than 20%. But interestingly, when you look at organic fertilizers and compost and you compare their release rates, there wasn't a difference in release rates. A little bit higher for organic fertilizers, but again, there's no difference. So the controlling factor of release rates is still not clear. And if you take the maximum, the peak amount that each of the fertilizers reach during the incubation period, we compare compost, manure, and organic fertilizers here, we again see that there's a strong trend with organic fertilizers and C to N ratio here. But then as you go towards compost, where you get in the lower C to N ratio, you see that that there is a less of a trend with C to N ratio there. So it seems like once you start to get up into the higher C to N ratio material, you get more carbon complexity and a different release rate of nitrogen. And it becomes less predictable. So we did this in the lab, and as you can see here, comparing soil types and then all the different fertilizers here, and then 2 different temperatures. And you can see that in general, the fertilizers acted differently across temperature ranges as well as across different soil types. So these trends keep coming out, and so the temperatures had a different impact across amendments, as well as different outcomes within soil types themselves. And soil types ended up having a limited effect on this total mineralization from the amendments. But the soil type altered the rate of release of this material. And initial immobilization was detected in both the aged chicken manure, was a bit surprising, as well as the alfalfa, loose leaf alfalfa, dried alfalfa material. So we took a subset of this and tried to answer this, or tried to understand the impact that these amendments have on this microbial carbon pool. And so we used microbial biomass carbon as the proxy for whether these amendments are going to increase the size of that pool, potentially increasing the outcome of storing more of these organic nutrients, or storing more nutrients in organic forms. And we found really that the original starting microbial biomass carbon in both of these soil types was different, although that but we found that the ending values, so the gray bars here are the after 60 days of incubation, and the black bars are the beginning of the incubation. We found that really we didn't find too much difference in the beginning and the outcome, beginning and the end of the incubation, meaning that the soil microbial biomass impact was limited from these low C to N ratio amendments. So if we don't try to look at this in the field, then we would be doing a disservice to the project here. So we used a 15N labeled tracer, and we added these amendments on top of it and saw, did it increase the, so we asked the question if nutrient use efficiency of the inorganic nitrogen pool was increased by adding these organic amendments. And we found indeed that, and this has been shown in different types of amendments, and we found it here that both pelletized chicken manure and compost increased the nitrogen uptake, total nitrogen uptake of the inorganic nitrogen pool. And so we also want to understand the total release from compost, and so we're using a mesh litter bag experiment, buried litter bag experiment. We're hoping to combine these to better understand what's going to become available from the pool, from the amendment itself, as well as how the amendments then impact the existing inorganic nitrogen pool. So conclusions and future directions. We found that C to N ratio appears to be a dominant control in N release from these organic fertilizers in particular, but not so much from higher carbon and higher C to N ratio amendments, and that temperature and soil type appear to have a varying effect. So it doesn't necessarily mean that if you're in one certain soil type that we can predict the outcome. Just because the impacts that temperature and soil type have on the N release rate does vary. And so, like I said, with the microbial biomass carbon, we found that there's minimal impacts of organic fertilizers on the microbial biomass carbon pool. And so if we think about this long-term nutrient planning as the changing, as adjusting for the amount that we are changing the organic nitrogen pool or the organic carbon pool, these materials might not have too much of an impact on that pool in the long run. So what you put down this season may last into next season and the next season, but it might not have long-term outcomes 5 to 10 years down the road. So hopefully we can take this information then and use it to use a biogeochemical model called DASCENT, which is the backbone of the Comet Farm Planner. If anybody's heard of that, it's a great tool. Check it out. And so hopefully we can kind of see how Comet Farm and DASCENT are projecting these long-term outcomes, and then maybe adjust them to help inform those models, or the DASCENT model better to accommodate these types of C to N, low C to N ratio amendments. And I just wanted to thank before we go into our survey, I just wanted to thank our project team. So Will Horwath is the PI, and my advisor, and he's given a lot of direction to the project. So I just wanted to thank him, as well as Shah Zubacher has been an important component of this project in guiding a lot of the incubations and a lot of the modeling work. And then on the ground, we have our UCA&R team led by Maria De La Fuente, County Director of Monterey, and many staff and student interns have helped supported this project. So thank you to all of them. And are we ready for the survey? Yeah, that's very interesting what we're seeing here. So that's good to see that people are planning annually and thinking about the next year's N availability, and some people are planning 5 years out. It'd be interesting to get their input and their thought process. So thank you everybody for responding to these polls. They'll be very helpful. And thank you, Aaron, for running them. All right, well, thank you, anyone. If you have questions, please reach out. If you'd like to be involved in a more in-depth survey, please reach out, and we can send that over to you. It takes a little bit longer, and we're trying to gain more information on your decision making process around nutrient planning in the long run. So I appreciate everybody's time, and I will pass it back over